Good morning, everyone. Today we will discuss about colorimetry and its principles. Aim is to study the principles of colorimetry. Colorimetry is the estimation of colored solute in a solution by comparing its colored intensity with that of a standard solution containing a known concentration of the same solute. The definition of standard solution means a solution of known concentration. Here the visible light of same intensity and identical wavelength is passed through both the solutions. Colorimeters. It is the instrument used to facilitate the exact matching of two colored solutions. Types of colorimetry. The first one is by comparison against a series of standard. The unknown colored solution is compared by inspection with a series of colored standard representing the substance being determined in known graded concentration. Here inspection means just observation by our eyes. Okay. There will be a series of standard. That means a standard. The color, we have to match the color with the standard, and then once the color is matched, then we can find out the concentration from the standard. The concentration of the unknown solution is given by concentration of the standard which it exactly matches. So this is how it's done. The next one is by duplication of colors. A relatively concentrated standard solution is measured into a blank containing the reagents as used in the sample until the color matches that of the sample. Okay. Here blank solution is the solution which contains all the components of a reaction or all the reagents except the analyte. Okay. So the volume of the standard solution required to prepare the duplicate is the measure of the test substance in the sample. Okay. Now next is by dilution to color match. Either the unknown or the standard is diluted with water or other solvents until the two colors exactly matches in intensity. It is done under similar condition of illumination or lighting, and the depth of the solution through which the light passes also should be same. Thus, if the unknown must be diluted twice the original volume to which matches a given standard, then the original concentration of the unknown is assumed to have been twice that of the standard. Okay, let's say the standard is like 2 mg per dl. Okay, then if we dilute the unknown twice its original volume, okay, to match the color with the standard, then we can say that the color the concentration of the unknown is like 4 mg per dl if the standard is 2 mg per dl. Okay, just like that. And the next one is visual colorimeters. They use visible mixed white light from an incident incandescent electric lamp. Components of a colorimeter. The first one is light source, a tungsten lamp emitting light in the visible range only. Here in colorimeter we use only visible range white light. We don't use ultraviolet or infrared. Filters. Filters are colored glass filters which absorb most of the light and permit light of the corresponding color only with sufficient narrow wavelength. Okay. So once the white light passes through the filter, only a single color will come out. That means only monochromatic light will come out after it passes through the filter. The color of the light which will come out depends on which filter we use. For its determination, a suitable wavelength of light is selected by using a specific filter of known wavelength. The use of different filters depend on the color of the resultant solution. So for each solution, for each color, they have different filter. So we have to find it. So the filter depends on the color of the solution. Complementary color filters are used because of the absorbance is maximum at the corresponding light. So if let's say if the color of the solution is like bluish green, then we can use red filter whose wavelength is 650 to 700 nanometer. And let's say if the color of the solution is like around like purple color, then we can use green filter which wavelength is 505 to 555. And let's say if the color of the solution is like yellowish blue, okay, the solution means the sample solution which we are about to measure the concentration. Okay, then for yellowish blue solution we can use filter violet filter whose wavelength is like around 350 to 430 nanometer. Okay, so this is how filters are chosen for the respective reaction. Qubits are glass tubes of usually 1 centimeter diameter and the uniform thickness in which the absorbance is measured. Qubits are the glass tube in which we will put the solution which we want to measure. It is optically transparent, thoroughly clean, and devoid of any scratches and free from any contamination. Okay. And it can be square, rectangular qubits with flat surfaces, or it can be cylindrical. But usually the diameter, if cylindrical, the diameter is like 1 centimeter, and even if it's square also, like the side is 1 centimeter, length of the side. The optical part always is usually 1 centimeter, but the capacity may vary. Glass qubits are used in visible range colorimetry, while quartz or fused silica qubits are used in measurement in UV range. Here colorimetry only visible range are used, but if you want, but in spectrometry or spectrophotometer, we can use UV range or UV light. Photosensitive detectors. They are the components of colorimeter which convert the light energy to electrical signals, which is proportional to the number of photons cracking the photosensitive area or surface. Measuring device. The photodetector response is measured by following readout devices like galvanometers, emitters, recorders, and digital readout. Signals are transmitted to the computer or printout devices. Most modern instruments are of direct reading type, where the amplified detector signal operates a galvanometer. Now here's the definition of a blank solution. A solution consisting of all the components of a reaction except the analyte. That is a blank solution. Now this is how the colorimetry works, colorimeter works. First, the light will come from the light source and it will go through the slit and it will pass through the lens, and this light is polychromatic light. It's polychromatic white light. Once it goes through the filter, it will be filtered out and only a monochromatic light of a single wavelength or a single color will come out. Now this light will pass through the cuvette. Cuvette is actually a glass tube containing the solution. Okay, it will pass through the cuvette and reach the photocell or the photodetector. The photodetector is connected to the readout device and it will give the absorbance or the optical density of the solution containing in the cuvette. Monochromatic light means light of single wavelength or single color. Photometry is based on two laws, Beer's law and Lambert's law. According to Beer's law, when a parallel beam of monochromatic light is passed through a solution, the absorbance or the optical density of the solution is directly proportional to the concentration of the substance in the solution. Okay, so if the concentration is increased or high, then the absorbance also will, or the optical density will increase. Okay, if its concentration is low, then the optical density or the absorbance also will decrease. So let's say A is the absorbance and C is the concentration. A is directly proportional to C, or we can write as A is equal to C Kc, where K is a constant. Now Lambert's law. Lambert's law is, according to this law, the absorbance or the optical density is directly proportional to the part of light which is the thickness of the solution. Thickness of the solution means, like in case of cuvette, it is diameter is 1 centimeter. So the light is passing through the cuvette. That means the thickness of the solution through which the light is passing is 1 centimeter. Okay, so that is the thickness of the solution. Now the absorbance or the optical density is directly proportional to the part of the light L. We can write as A is equal to KL, where K is a constant. Now combining the two laws, we can write absorbance is equal to KCl. C is the concentration, L is the length of the path through which the light passes through the solution. So where A is equal to KCl, where K is a proportionality constant. It is also known as extinction coefficient or absorption coefficient. It is fixed for a given substance at a specific concentration and wavelength. Now Beer's law can be applied or obeyed only in dilute solutions. It cannot be applied in concentrated, highly concentrated solution, okay, where the light cannot pass through. In routine colorimetry, absorption coefficient is not used. Instead, concentration of the unknown is calculated by comparison of its absorbance with the absorbance of the solution of known concentration. Now we'll go to this deduction part. If Ct is the concentration of the test sample and Cs is the concentration of the standard, then the absorbance of the sample AT will be KCTL, and the absorbance of the standard will be, the absorbance of the standard AS will be equal to KCSL. CS is the concentration of the standard. Now for the substance at a constant path length under same conditions, K and L are constant. K is constant and L is constant. And L is constant. We are taking L as constant because let's say in the cubit the diameter is 1 centimeter, so it's constant for both of them. So we will take K and L are constant. Then in this case, the absorbance of the test sample will be equal to the concentration of the test sample, and the absorbance of the standard will be equal to the concentration of the standard. From these two we can write AT by AS is equal to CT by CS. Then concentration of the test is equal to, that is CT, is equal to AT by AS into CS. As the concentration increases linearly, the absorbance increases linearly, that is proportionally. As the concentration increases linearly, the transmittant decreases logarithmically. Now, uses of colorimetry. Here it can be used for estimation of blood sugar by Nelson-Somoyogi method and estimation of serum protein by Biuret method, and it can also be used for estimation of urea by diacetyl monoxime method, and for serum creatinine by Brod-Sirota method, serum bilirubin by Vandenberg method, and serum cholesterol by Sekel's method. So from this formula we can find out the concentration of the unknown solution or the test sample by knowing or finding out the absorbance of the test and the absorbance of the standard. Okay, and the concentration of the standard is already known, so don't need to find out. So once we measure the absorbance of the test and absorbance of the standard, then we can know the concentration of the test solution. And this formula will be used to find out all the estimation of all this blood sugar, protein, urea, creatinine, bilirubin, and cholesterol. That's why colorimetry is very important. Okay, thank you everyone. I hope you understand the topic. Now we will proceed to the next video for demonstration.